Okay, so we're going to do this just like yesterday. And I'll start with like a very high-level introduction to like a 10-minute or so of the analysis resource. And then afterwards, we're going to have for every single one of the topics I'm touching on a much more detailed presentation, how to use it, etc. So as you see here, obviously today is the tools day, the analysis resource day, which is essentially a compendium of predictive and analysis tools that we've generated using the data in the IEDB. And I want to start here acknowledging the people involved in it. Many of them are in this room, and we desperately need an updated picture. Some of these are no longer with us. What we also really want to stress is that all of the tools, or many of the tools, are developed in collaboration with the Danish group from led by Morten Nielsen. We have actually two students that are with them visiting here as well. Ole Lund and Søren Bus also in Denmark. And then, yes, you know, we already talked about the collaboration with Kevin and also Ralph, who works as well. So, as I mentioned briefly, so we have this distinction between prediction tools and analysis tools. So the idea of prediction tools is that they generalize the kind of data that we have in the database to allow machine learning algorithms to predict, say, binding to MHC class 1 molecules or processing, predict what regions in the protein are accessible to antibody binding. And then as a contrast, we have the analysis tools. So this is the kind of tools that if you are into epitope research and you're developing, say, using an assay to discover a set of epitopes and you want to learn something about them, or you perform queries and you want to learn something about the epitopes that are retrieved, you can analyze things like the conservation of these epitopes or the projected population coverage based on HLA frequencies and so on. And I'll talk about these in some more detail. So epitope prediction tools, as I said, like we are deploying in machine learning here, and I want to explain that on a quite simple example, or a comparatively simple example of MHC class 1 binding. So in an MHC class 1 binding assay, we have purified peptides that are measured for their affinity to purified MHC molecules, at least the most common assay used, where essentially you have a known high affinity binder that you label with something. So typically, actually, you use iodine labeling, and then you come asking how your peptide of interest, when you titrate it in, at what point you outcompete 50% of the binding of your reference ligand. So then you're getting an IC50 value, so inhibitory concentration 50%, which if you perform this assay under the right conditions, then the IC50 logarithm of that is roughly proportional to binding free energy. So a low IC50 value means that you need a little amount of your peptide of interest to outcompete this, so this would be a high affinity binder, which always is confusing when you have a low number and say this is high. So that's IC50 values, and you can measure these for a bunch of peptides, and you can say, well, that solves my problem. I can just measure all the affinity for all the peptides in a given, say, virus, and then you know what the potential binders are. But the problem, of course, is that is impossible, as in just peptide synthesis cost will kill you. So what we're trying to do instead is...
What about the units of IC50, nanomolar or?
Nanomolar, yeah.
Nanomolar.
In this case, yes. So what we want to do instead is kind of learn the pattern of what peptides bind, and that is what machine learning is. Essentially, it means that you're trying to identify some kind of function, which, given the sequence, gives an output that approximates the affinity. And there's a large number of different approaches: neural networks, support vector machines, hidden Markov models, linear programming, etc., all of which are essentially machine learning approaches. And all they really do is, what they differ in is what do they mean by find? How do you find the function? So in the case of neural networks, for example, is like you propagate, whatever, back propagation or something like that. Or least square optimization for some of the linear programming things. And what is the function space you're looking in, and how do you determine if this approximation of the values you get out, if that is acceptable? So one simple example of how this can be done is actually having as your function, as your model, a matrix where for every residue in a peptide, say this is a 9-residue peptide, you have a value of how different residues at that position contribute to binding. And for a given peptide, you take all these values, you sum them up, you add a constant, and you get a prediction back. And actually, this simple approach for MHC class 1 peptide binding already works quite well. And then you essentially train it by saying, given all these known affinities that I have for known peptide sequences, how do I optimize these matrix coefficients to minimize the distancing? So one of the things we do a lot of is trying to evaluate the prediction quality, to compare these different approaches, given that there's essentially weekly still new algorithms coming out that are obviously always claiming that they're better than...
I'm sorry, could you go back to the previous slide? Your matrix. This is an unsymmetrical matrix. What's the 1 to 9 across the top?
Oh, sorry. This is position in the peptide. Position 1, 2, 3, 4, 5, 6, 7, 8, 9. This is 9 rescue peptide. So in this case, position 9 for binding for AO201 has the typical preference for hydrophobic residues, etc. Okay. Sorry, I should have said that. Yes, evaluating prediction quality. So we distinguish between evaluating external predictions. So the first, there's a number of web servers out there where we essentially retrieve for a set of peptides then that we have known measured affinities for. You see the measured affinity, each dot is one peptide. You have classic servers such as SIFPIDE give you a prediction score, which is based on essentially the overlap of that peptide sequence to a known motif associated with a given allele. And then you can plot here, and you see that there's a clear correlation between the predicted score and the measured affinities. The next, actually most classic method is this BIMES that was developed in 1994, which does a linear regression essentially, and which predicts half-life of binding. And you see there also is a correlation with the measured affinity. And then you can evaluate such quality of these different predictions by an ROC analysis. That essentially means you're trying to separate out what we know to be good binders of biological meaningful binders. That is typically a threshold associated with that of less than 500 nanomolar. So you want to identify the peptides that fall here using these scores. So essentially for a given cutoff of your score, you're getting true positive predictions. So those are the better than your score. You also have false positives that are better than your score but don't bind with less than 500 nanomolar, etc. You can do this systematically for all kinds of thresholds in your score, and you get an ROC plot which where the area under this curve or the AUC value evaluates prediction quality. And so this was kind of the first thing how we started off with, and what we found very quickly is that yes, you can compare these different external methods, but does that really tell you something about which method is better, or does it tell you something about the data that was available to the methods? So that is one of the problems. So we also implemented a large number of different methods internally and then trained them on the same data and then compared how the different algorithms perform in this setting. And for that we use cross-validation, where essentially you take the whole data set, you take out a slice, like 1/5 of your data, you train your methods on 4 of the remaining 5, you predict the ones that you left out, and you're asking how well does that prediction work. So and then you can do essentially the same thing as we did before, use an ROC analysis, calculate AUC values to evaluate prediction quality. And the results are going to be the upcoming talks. So we have a number of prediction tools available in the analysis resource for B-cell epitope predictions, and again, this is going to be covered in much more detail in the next talks. We have implemented classic ways of how this has been done in the past based on amino acid scales, such as hydrophilicity. We've also added a number of machine learning approaches and structure-based approaches. These are thought to be organism unspecific, so antibodies are thought to essentially work similarly, independent on what the host is that makes them. The accuracy tends to be rather poor, and the evaluation is also rather tricky, as the kinds of assays that you're using to generate your antibody epitope data can be quite divergent. For MHC class 1 binding, we have a broad breadth of more than 80 alleles for which specific predictions are available. We also have non-human primate, non-primate ones. And then pioneered by the Copenhagen group from Morton is the concept of pan predictions, of actually using a neural network that learns in parallel the sequences of the alleles it makes predictions for, the peptides that bind to it, and then can extrapolate for a new MHC allele with a sequence that it hasn't actually specifically seen before, how that would bind to peptides, and then it works quite amazingly well and provides you essentially a coverage for every human MHC allele out there. These have very high accuracy and highest performance in comparison to MHC class 2 binding and antibody epitope prediction with average AUC values clearly greater than 0.9. And there has been essentially more or less incremental updates in performance being done since 2006. For MHC class 2, we have much fewer data points available. There's 24 amino MHC alleles where we have good binding data sets available. We have limited non-human data, which is all mouse so far. And we have, again, using the pan prediction concept from Morton, prediction data that cover all the human alleles, but nothing like that exists currently for mice or other species. The accuracy is definitely lower than before. It's the last larger comparison we did, we had AUC ranks of 0.87, but that is substantially improved from where it was in 2006. 2006 is the comparison point where we started with the IEDB. So this is still an area where definitely further improvements are possible and necessary. Then we have MHC class 1 processing predictions, which are incorporating other factors than MHC binding, such as proteasomal cleavage and tap transport. And we have predictors trained on eluded ligands that are essentially using data from peptides that are naturally processed, presented, and bound by MHC molecules, which essentially combine the effects of these different processing steps in the peptide sequences that we find in these eluded ligands. So these processing approaches are so far only available and validated for humans, and they do provide improved predictions over MHC binding predictions alone. And this is, while rather small, it's definitely statistically significant. And actually, there are several large-scale validations out there, but specifically the question how well they predict T-cell epitopes is still a little unclear. So epitope analysis tools, those are again the ones where we use them to have existing data sets and analyze them more. And again, I'll talk about those later. So the different things that we're looking at is how conserved is a set of epitopes in a set of protein sequences. Cluster analysis, like do you have homologs in your epitopes that you're looking at? Population coverage, this is the projected. If you have MHC restrictions for your epitopes and you have a set of epitopes, what is going to be the response frequency in the human population? Then we have the EpiFilter tool, which is meant to essentially tidy up the kind of data you're getting from an IEDB query, focusing on the kinds of epitopes that have the more meaningful cellular immunology data and better support to develop peptide epitope pools. The rate tool that infers HLA restriction. If you have a set of epitopes and a reactivity pattern in donors that are HLA typed. A de-immunization tool that is essentially looking for a dominant region based on MHC class 2 binding and suggests amino acid substitutions that create non-immunogenic versions of this. And again, like all of these are going to be covered in more detail. I'm running out of time. That's why I'm speeding up and talking. So to summarize this again, like we have these prediction tools that extrapolate from the data we have in the database to essentially identify new epitope candidates, which could then be tested experimentally, and we have analysis tools to help examine existing sets of epitopes to gain new knowledge. And as I'm running over time, I would prefer to skip to the next talks that are going to go over.
So all the tools will go individually through, so we can do all the questions as that goes. And next we have Sandeep, I believe. Oh, perfect. Okay.